Howdy! Welcome to Chapter 7, Microbial Genetics 1. This is an overview of microbial genetics. So, if we want to talk about genetics, we have to talk about the genomes. So we have to talk about chromosomes, we have to talk about chromosomes, we have to talk about DNA. So, in this cartoon, here's letter A. You probably remember this from high school or from college. DNA has the base pairs A, T, C, and G. In DNA, A and T, when they're in the double helix, so down here, when they're in the double helix of DNA, then A will always pair with T, and G will always pair with C. And can you guess what these dots mean? Well, if you said the dots between A and T are hydrogen bonds, then you're right. Then you listened to my previous videos. So those are hydrogen bonds. When A pairs with T and C pairs with G, they form bonds, the hydrogen bonds form, and you get, and then there's other forces, but you get the structure called the double helix, the DNA double helix. And then, so that's in DNA. In RNA, there is no T. So in RNA, instead of a T, you have a U. And then the U will pair with A in RNA. In RNA, the U will pair with A. Yeah, so uracil pairs with adenine in RNA. And then, so in this cartoon, they show this purple strip here. So that's the sugar phosphate backbone in DNA, the sugar phosphate backbone. So here's the sugar, and here's the phosphate, and then here's the bases, guanine pairing with cytosine with the hydrogen bonds. So that's kind of an overview of the structure of nucleic acids. So let's look at the definition of a genome. So a genome is the entire genetic complement of an organism. Includes the genes, the nucleotide sequences, that's like A, T, C, G, A, G, 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 whatever the sequence is, and then these things called transposons. So transposons are, in this cartoon, transposable element shows a bacterial chromosome, which is just a single circular chromosome that's double-stranded, and a piece of that chromosome, this pink stuff, will be inserted into a plasmid. That's called a transposable element. Structure and replication of genomes for prokaryotes, prokaryotic chromosomes. That's where you see most of the DNA. So if you look here, this is a transmission electron micrograph that's colorized. This is the nucleoid, that's where the chromosome is. This on the bottom is a scanning electron micrograph, and here's the bacterium. The cell, so the bacterial cell was broken open, and here's the chromosome. So all this kind of back and forth stuff. This is a single chromosome that's circular, and then also what leaked out of the bacterial cell was this thing called a plasmid. So the prokaryotic chromosome, which is this big thing that's spread all over the place, is the main portion of DNA, and then there's also proteins associated with the chromosome. There's RNA associated with the chromosome. And you have a single chromosome copy, so that means you just have one single circular chromosome in bacteria, but it's double-stranded. In addition to the chromosome, there's more DNA contained in these things called plasmids. So a little bit more about plasmids. So again, here's the plasmid. This right here could also be a plasmid as well. So plasmids are small molecules of DNA that replicate independently of the bacterial chromosome. Plasmids, they're not essential for normal metabolism, growth, or reproduction. They're used for other things, but they can, the plasmids in these bacteria can confer survival advantages. So there could be genes on this plasmid that will help the bacteria fight off antimicrobial drugs that we might try to kill the bacteria with. There's a whole bit different types of plasmids. There's fertility factors. They carry instructions for conjugation. So conjugation pilus allows one bacteria to transfer a plasmid to another bacteria. A plasmid could be contain resistance factors. Those that would be DNA that carries instructions for drug resistance. Bacteriocin factors in a plasmid would carry instructions to kill other bacteria. Virulence plasmids, they carry instructions for pathogenicity, so to make people sick, basically. This cartoon shows the bidirectionality of DNA replication in prokaryotes. So this would be a bacterial chromosome. It's double-stranded, as you can see. Here's the origin of replication. So this green part here is where replication begins. And you get replication in both directions. So this dark purple thing is the parental strand, and then this lighter purple strand is the daughter strand being made from the parental strand. But as the replication forks move outward, and then you terminate replication, and look, now you've got two new bacterial chromosomes. And we've talked about this. The bacterial chromosome is one long single molecule of double-stranded helical supercoiled DNA. In most bacteria, the two ends of the double-stranded DNA, they bond together to form a physical circle and a genetic circle. So this is the cartoon showing the central dogma of genetics, or the central dogma of biology. Let's look at the top. Here's DNA. DNA is double-stranded, and here's the nucleotide sequence: A, T, G, C, G, G, T, A, C, T, T, A. That's the nucleotide sequence. And then here's the complementary base pair. So T always binds with A, remember, and G always hydrogen bonds to C. So when it's time to make some proteins, first thing you have to do is you have to transcribe DNA into messenger RNA. So the strand that you read from DNA, that you transcribe from DNA, would be this strand here. So you transcribe the T to an A in the messenger RNA, and then from the A in the DNA, you transcribe to a U, because remember there's no T in messenger RNA, and then the C would be transcribed to a G, etc. And then you'd have this messenger RNA transcript. That's what they call it, a messenger RNA transcript. And then that messenger RNA transcript would get translated into a protein on a ribosome. So you take this messenger RNA transcript and you put it on a ribosome, and then the ribosome would hook up these amino acids, methionine, arginine, tyrosine, and leucine. You'd make a polypeptide on the ribosome, and then you'd hook your polypeptides together and make a protein, and that'll give you the phenotype. And that's basically, the phenotype is what you see as the organism, what it looks like. So, for example, the phenotype of my eyes could be, blue or green or brown. That would be a phenotype. So these are the details which you don't really have to know for the test, but in the top cartoon, this is DNA, the double helix, and when you're ready to transcribe DNA into RNA, you need an RNA polymerase. So poly, making a polymer ase, so it's an enzyme. So the RNA polymerase attaches to DNA. It finds a promoter region in the DNA because that's where the gene starts. And then the RNA polymerase moves along the DNA. So transcribing from the DNA template, the RNA polymerase transcribes the RNA strand, which is right here. And then at some point, there's a terminator sequence in the DNA, so the RNA transcript is done being made and it's released. So this is a slide talking about the genetic code. It's a triplet code. So if you look at this at the top, this is the RNA transcript, but you could also call it the RNA codon, because the RNA codon is a triplet code that will be read on the ribosome to make a protein out of amino acids. So for example, here's the RNA transcript or the RNA codon. This first part is AUG. So the first letter is A, and so you come over here, you find the first letter, the first nucleotide is A, and then the second nucleotide here is U. So the second nucleotide, so then you read the second nucleotide up here, so the second nucleotide is U, and then the third nucleotide it's a triplet code, so you read it 3 at a time, is G. So you come over here, here's the third base in this sequence. So here's a G, and you see this is AUG. So you come over here, AUG, and you read start. So AUG is the start codon to start translation of the messenger RNA sequence. So DNA codes for a protein by transcribing a messenger RNA codon. So this whole thing is a messenger RNA codon intermediary, which is translated into a protein. Protein is a string of amino acids. So for example, a Vibrio cholerae obtained a gene that codes for cholera toxin that makes us sick. Okay, so let's look a little bit more on this genetic code. If you, well, let's read the second part here. CGG, so the first letter in the codon in this second triplet is C. So the first nucleotide is C. The second nucleotide is G. So the second nucleotide over here is G. And then the third nucleotide is also G. So it's CGG. So you come over here, find CGG, and that codes for the amino acid arginine. So at this triplet code, when you're out on the ribosome, you will put an amino acid called arginine that would correspond to this CGG triplet code. So this is just an overview of the ribosomes. So remember, DNA gets transcribed into RNA, and then RNA gets translated into a protein on a ribosome. And this just shows what the ribosome is made up of. We talked about this before. The prokaryotic ribosome is called a 70S ribosome, and the eukaryotic ribosome is called an 80S ribosome. Okay, so here's the ribosome. Now remember, the messenger RNA gets read on the ribosome, or translated. So if we look down here, here's our messenger RNA. The ribosome's going to move this way, and the messenger RNA is going to move this way. And each one of those triplet codons on the messenger RNA is going to be read and associated with an amino acid. This is a little more detail of what the ribosome looks like. So it's got these EPA sites. So the A site is an arrival, here's a processing site, and here's an exit site for the growing polypeptide. So here is our ribosome, 5' to 3'. This is our messenger RNA transcript, and then here's the codon. So remember, AUG here means start, and when you start making a protein from amino acids, the first amino acid that you use is F-met, or methionine. And this blue thing is the transfer RNA molecule that binds to the codon with this anticodon part. So the transfer RNA has an anticodon that binds to the codon on the messenger RNA, and the transfer RNA brings in the amino acid. So remember, the messenger RNA is moving this way, and the ribosome is moving this way, and each one of these blue things is a transfer RNA that's going to have an anticodon here that will read the codon on the messenger RNA and bring in the next amino acid. And then between the two amino acids, the peptide bond will form. So that's the processing. The peptide bond will form when these two guys are in the P and the A site. And then you can see that the messenger RNA moves one way, the ribosome moves the next way, and then these transfer RNA molecules eventually will exit. And down here you can see now we've got 3 amino acids linked together by a peptide bond. Here we've got 1, 2, 3, 4, 5 amino acids linked together by a peptide bond. And then here comes the next transfer RNA, bringing alanine, the amino acid alanine, and this transfer RNA will arrive in the acceptor site. And then the whole thing will move, you'll form a peptide bond, and eventually your polypeptide will form. And then the messenger RNA will fall off the ribosome, and you will have made your polypeptide. In a prokaryotic cell, you have a polyribosome, so you've got ribosomes translating proteins all along the messenger RNA. Regulation of genetic expression. So how does a cell know to turn on a gene, make a transcript, and then translate that messenger RNA into a protein? 75% of genes are expressed at all times. Other genes are transcribed and translated only when the cells need those proteins. So that allows the cell to conserve energy. And I think that's the end.